The overall goal of this procedure is to generate neural crest progenitor cells from human pluripotent stem cells in a short, reproducible, and defined protocol. These cells can then be used for either the study of neural crest-related diseases in vitro or the continued differentiation of these cells into progeny cells. This is accomplished by first plating a high density of human pluripotent stem cells in a monolayer on matrigel. The second step is to differentiate the cells into neuroepithelial cells using a defined sequence and combination of small molecules that affect key signaling pathways. Next, the neuroepithelial cells are replated and further differentiated into neural crest progenitor cells. The final step is to isolate the neural crest cells using fluorescence-activated cell sorting. Ultimately, immunofluorescence and functional assays, such as the cell's ability to migrate, are used to show that proper neural crest cells were generated.
The main advantage of this technique over existing protocols for the derivation of neural crest cells from human pluripotent stem cells is that it is shorter, it's only 18 days, it is more reproducible due to the lack of MS5 feeder cells, it's easy to upscale it, and it's highly reproducible among human pluripotent stem cell lines.
The implications of this technique extend towards studies of the mechanisms of human diseases related to the neural crest lineage and show potential for improving detailed in vitro modeling and drug screening of these diseases.
Prior to starting this protocol, ensure that the colonies of human pluripotent stem cells are large enough to be seen by eye and still have sharp edges with few differentiating cells at their borders. Then, when the hPSCs are ready to be split, aspirate the medium, wash once with 5 milliliters of 1-xPBS.